For several decades in the mid to late 20th century, niacin was one of the most respected cardiovascular drugs in clinical medicine. It was among the first compounds shown to modify blood lipids in a therapeutically meaningful way, and the Coronary Drug Project, a massive U.S. government-funded trial completed in the 1970s, demonstrated that niacin reduced non-fatal heart attacks and lowered mortality even years after the trial ended. This was landmark evidence. Niacin was prescribed widely, studied extensively, and considered a cornerstone of lipid management for patients who couldn't tolerate or didn't respond adequately to other interventions. Then two major trials, AMHI, published in 2011, and HPS-2, Thrive, published in 2014, reported that adding extended-release niacin to statin therapy did not reduce cardiovascular events compared to statin alone, and that HPS-2, Thrive in particular showed a small but real increase in certain adverse events in the niacin plus statin group. The medical community responded to these findings with significant niacin skepticism. Guidelines were revised. Prescriptions dropped. The narrative flipped from niacin is a proven cardiovascular tool to niacin doesn't work and may cause harm, with a speed that outran the nuance of what the trials actually showed. The trial design issues are substantial and underappreciated outside specialized cardiology. Both AMHI and HPS-2, Thrive studied niacin as an add-on to statins in patients whose LDL was already tightly controlled. When LDL is already at target through statin therapy, there's less remaining lipid-driven cardiovascular risk for niacin to address. The trials were essentially asking whether niacin could improve on an already well-managed baseline, a different and much harder question than whether niacin works in statin-naive patients or patients who cannot achieve LDL targets with statins alone. HPS-2, Thrive used a formulation of extended-release niacin combined with laropiprant, a drug added specifically to reduce flushing. Laropiprant has its own biological activity beyond flush suppression, including effects on platelet function and prostaglandin pathways. The adverse events observed in that trial may have been partially attributable to laropiprant rather than niacin itself. Laropiprant was subsequently withdrawn from markets in several countries for safety reasons. Using a trial studying niacin plus laropiprant as evidence about niacin alone is analytically unsound. The damage to niacin's reputation has been significant and arguably unjustified by what the evidence actually shows. Niacin's HDL-raising effects remain unique in pharmacology. Its NAD+, boosting effects remain real and potentially important for aging biology. Its GPR109A mediated anti-inflammatory effects are real and underexplored. The compound that cured pellagra demonstrated cardiovascular benefit in monotherapy trials and boosts the same cellular machinery that an entire longevity supplement industry is built around deserves a more careful reassessment than it has received.